Good day, everyone. Thanks for attending this talk. My name is Shahin Nasr, and I'm presenting our study about differential mechanisms of absolute versus relative disparity encoding in human extrastriate visual cortex and impacts of amblyopia on them. As disclosures, Dr. Bex has financial interest in adaptive sensory technology and a patent with Mass Eye and Ear and Northeastern University, and Dr. Hunter has financial interest in luminopia and Ribion and a patent with Boston Children's Hospital. The rest of authors have no commercial relationship to disclose. Horizontal disparity between inputs from the two eyes is one of the main cues in depth perception. Disparity cues are divided into relative versus absolute. Relative disparity is an important cue in binocular depth encoding. Relative disparity cues are believed to be encoded by V2 tick stripes and tick-type columns in subsequent visual areas. Absolute disparity, which refers to the disparity measured relative to the fixation point, is used for vergence control. Absolute disparity cues evoke a strong response in the motion selective area MT, but the contribution of earlier visual areas in their encoding is not clear. Development of neuronal mechanisms underlying disparity encoding relies on normal binocular vision in early stages of life. Disruption of binocular vision in this critical period leads to amblyopia, which in most cases decreases stereo acuity. Animal models of amblyopia suggest that amblyopia is associated with a decrease in the number of stereoselective neurons. However, direct evidence for this phenomenon in humans is rare due to the small size of stereoselective sites in human visual cortex compared to the spatial resolution of neuroimaging techniques used in human studies. It's also not clear whether amblyopia has the same impacts on absolute and relative disparity encoding. This study consists of two parts, both of them based on using high-resolution fMRI collected in a 7T scanner, which gave us a nominal resolution of 1 millimeter. In the first part, subjects were scanned 3 times, during which mesoscale stereo, motion, and color-selective sites across extrastriate visual cortex were localized. These sites comprise our regions of interest. Reliability of our approach to localize these sites has been shown in our multiple previous publications. In the second part, each subject was scanned one more time, during which we measured the evoked activity in our regions of interest by stimuli with varying absolute versus relative disparity. To this date, 27 individuals have participated in our study. Among them, 12 were amblyopic, including 7 individuals with strabismus and 5 individuals with anisometropia. Controls were age-matched to amblyopes. They showed normal or corrected to normal visual acuity in both eyes and significantly better stereo acuity compared to amblyopes based on RANDOT test. We localized the stereoselective sites in our subjects by contrasting the response evoked by 3D versus 2D stimuli generated by random dot stereograms RDS. Subjects saw the stimuli through red and green filters that were installed on the head coil. 3D stimuli illustrated a checkerboard pattern in which surfaces moved asynchronously in depths. The level of relative disparity varied between -0.4 to +0.4 visual angles. 2D stimuli were based on the same pattern, but here the amount of disparity between dots remained 0. We localized the motion selective sites in our subjects by contrasting the response evoked by moving versus stationary concentric rings. Among these sites, motion selective area MT was expected to show a strong selectivity for absolute disparity. However, response of the other motion selective sites to either absolute or relative disparity cues were not clear. We localize the color selective sites in our subjects based on contrasting the response evoked by color varying versus luminance varying stimuli. These sites were used to better define the borders of stereo and motion selective sites, as we have shown in our previous publications. Activity measured in these sites were also used as a control, mainly because we did not expect any strong stereoselective response in them. As we mentioned, in a separate scan session, we measured the activity evoked by relative and absolute disparity varying stimuli in the regions of interest. Subjects were presented with 4 different stimulus conditions across different blocks. In 25% of blocks, stimuli were asynchronous disparity varying RDS that contained strong relative disparity cues. In the other 25% of blocks, stimuli were uniform disparity varying RDS that contained absolute but not relative disparity cues. In the other 2 block types, we presented the corresponding zero disparity conditions for these stimuli. Here you can see the location of those cortical sites that show a significantly stronger response to 3D versus 2D stimuli in one subject. Red to yellow colors indicate the p-value of 10 power minus 2 to 10 power minus 6. You can see the stripy pattern of activity in area V2 that extend to area V3, V4, and also area V3A. Beyond the retinotopic area, you can see activity in area MT and also in object selective area LOC. Here you can see the motion selective sites in the same subject. In macro level, you can also see a strong activity at the tip of middle temporal sulcus that we use to localize area MT. This motion selective activity extends posteriorly toward area V3A. Again, in finer scales, you can see the stripy activity pattern in V2, V3, and also in area V4. And then the color selective activity that starts in area V1 and extends toward area V2 and V3 and also V4. And there is almost no activity in area V3A. When we tested the V2 response to relative versus absolute disparity cues, we found a strong activity evoked within the stereoselective sites by relative but not absolute disparity cues. This phenomenon was similarly found in the central and peripheral visual field representations. Notably, in this analysis, all activities are normalized relative to their corresponding zero disparity condition. But from V3, things started to change. Here, especially in the central visual field representation, we found a significant response to absolute disparity cues in stereo and also in motion selective clusters. Interestingly, in motion selective clusters, this activity was stronger than the activity evoked by the relative disparity cues. In area V3A, this effect is equally apparent in the central and peripheral visual field representations. In both parts, motion selective sites responded more strongly to the absolute compared to relative disparity cues. An opposite pattern was detected within the stereoselective sites. And as expected from the previous studies in non-human primates, we found a strong bias in favor of the absolute compared to the relative disparity cues in area MT. But what happens in amblyopic individuals? Here you can see the response evoked by 3D versus 2D stimuli in one amblyopic subject with no measurable stereoacuity. As you can see, the stereoselective sites are simply non-existent in this individual. This is something that we see in most amblyopes that show either stereo blindness or worse than 200 arc seconds stereoacuity. Here you can see the motion selective activity in the same individual. Similar to the control subject that I showed, you can see area MT, extension of activity to area V3A, and a stripy activity pattern in V2, V3, and even in V4. Here you can see activity evoked by color versus luminance varying stimuli. Again, similar to the control subject that I showed, you can see activity in V1, V2, V3, and V4. Here again, you see almost no or very weak color selective activity in area V3A. As you can see here in area V3, the impacts of amblyopia are not limited to the encoding of relative disparity cues and/or the response measured within the stereoselective sites. Rather, amblyopes also showed weakened response to the absolute disparity cues evoked within motion-selective sites. Also here in area V3A, also here in area MT. So the take-home messages are: relative and absolute disparity cues evoke different response patterns within motion and stereoselective sites. Most importantly, our results suggest that the absolute disparity encoding is not limited to area MT. Rather, it can be found in the earlier motion-selective sites that are distributed within areas V3 and V3A. Our results also suggest that amblyopia affects encoding of relative and absolute disparity cues. This is consistent with the stereoacuity and Burgess control impairment in amblyopic individuals. Despite these findings, we are not still able to conclude whether these two impairments are correlated with each other or not. More data and tests are required before any conclusion. In case you are interested in the other impacts of amblyopia on the mesoscale functional organization of human extrastriate visual cortex, please come to our other talk during which Bryan will talk about this topic. Here we want to express our gratitude to our participants, collaborators, and funding sponsors, and thank you for your attention.